Okay, well, now we will continue from previous part. This is an extension from chapter 3, Physiology of Muscle. So, muscle contraction, the sliding filament hypothesis. So, to understand the sliding filament hypothesis, first place your right palm on the back of your left hand. Then now slide your right palm toward left elbow. So what happened to the distance between your elbows? It will be short. So this is how muscle contraction occurs. So the thin filament slide over thick filament. So this pulls the discs close together, or sarcomers close together. So when all sarcomers in fiber do this, the entire fiber gets shorter, which pulls on the endomysium, premiysium, epimysium, then attached to tendon, then pulls the bone. So when the bone is pulled, movement will be occurred. So this is known as sliding filament hypothesis. So this is actin, this is myosin. So actin is or thin filament slide over thick filament. So another alternative is this one. So this one is thin filament. So this the blue one is thick filament. So thin filament is slide over thick filament. So this tells us how movement occurs. So this between two Z discs is known as sarcoma. So from this one up to this one is A band. So this is sliding. This is how sliding filaments will occur, or how movement will occur between the thin and thick filaments. So now, as you know, sliding filament hypothesis. Here is what happens as filaments of slide and sarcomer and the muscle fiber shortens. So in the process of contraction, what happens to Z disc, or the distance between Z disc? It will be shortened. The length of A band, the length of H zone, and the length of I band. So the very important concept you should know is actin and myosin do not change the length during muscle contraction. Rather, actin filament slide over myosin filament. So actin filament or thin filament slide over myosin filament, but actin and myosin themselves does not change their lengths. So actin and myosin does not shorten or lengthen by itself. Rather, they will slide one over the other. So So sliding filament, all sarcomers in the fiber will contract together. This contract the fiber itself. So the number of fibers contracting will determine the force of contraction of the whole muscle. So if many fibers or the whole fiber is participating in contracting, the force of contraction will be strong. But few fibers are contracting, so the force of contraction will be weak. So we can actually divide the whole process of muscle contraction into 4 steps. The one is excitation step or excitation phase. The other is excitation contraction coupling, and the other is contraction, and the last is relaxation. So the muscle contraction process started with excitation and end with relaxation. So any movement is follows these steps, and any movement is having contraction, they will contract and they will relax. So the first step is excitation. In general, each muscle is served by one nerve from spinal cord. So one nerve is arised from spinal cord and will go to the muscle. So there is a word or a term which is motor unit. Motor unit is defined as a somatic motor neuron, and all skeletal muscle fiber it innervates is called the motor unit. Let me show you. This is muscle. This is one motor neuron which has arised from spinal cord, and this motor neuron will divide into different telodendria. So this area or telodendria will have other synaptic nodes here indicate synaptic nodes here is indicate. So this, by the way, this nerve plus this muscle in the whole. So somatic motor nerve plus this skeletal muscle, or this skeletal muscle, it innervates in together, is known as motor unit. So this circle is known as motor unit. So this is fibers, this is neuromuscular junction, a synapse between the neuron and this muscle. This contact area, synapse area, is known as neuromuscular junction. So our concern will be neuromuscular junction. So when this neuron is stimulated, all muscle fibers synapse upon will be stimulated and will contract as in it. So when there is impulses comes to this nerve or stimulated, so muscle fibers will go into synapse and stimulated the muscle contraction or stimulated this muscle region. So within the muscle, each axon go its own way. Within the muscle also, each axon having subplace each muscle fiber. So each axon will go its own way and eventually branch into multiple small extensions. So this is multiple small extensions. This multiple small extension is known as telodendria. So each telodendrium ends in bulbous swelling known as synaptic end bulb. So this area is known as synaptic end nerve. This is a vesicle which containing impulse of this telodendria. So this is synaptic end bulb, and the site of interaction between neuron and any other cell is known as synapse. But the site of interaction between neuron and muscle is known as neuromuscular junction. So the neuromuscular junction is a site of interaction between neuron and any other cell. So this is synapse. Synapse between neuron and muscle is known as neuromuscular junction. So there is a space, there is small space between synaptic end bulb and the sarcolemma of the muscle. This is known as synaptic cleft. The space between the sarcolemma of the muscle and synaptic end bulb of the nerve is known as synaptic cleft. There is depression in the sarcolemma. So in the cell membrane of the muscle there is depression. So this depression area is known as motor end plate. So motor end plate is a depression area of the muscle membrane that is used for synapse formation. So this is known as motor end plate, and the synaptic end bulb is filled with vesicles that contain neurotransmitter. So this neurotransmitter is known as acetylcholine. So acetylcholine is a neurotransmitter of muscle. So they are very important. So the motor end plate is chock full of acetylcholine receptor. So motor end plate means that is found and that's a depression on the and the sarcolemma of the muscle. So when since the neuron is contain, or synaptic end bulb is contain acetylcholine, so the motor end plate of the muscle contains also receptors, acetylcholine receptor. This important receptor is known as nicotinic receptor. So nicotinic receptor responds for acetylcholine stimuli or synapse generation or potential. So this is a neuron. So this is a neuron. So this is synaptic vesicles that is containing acetylcholine, this blue. So this is acetylcholine with synaptic vesicle. So this is a mitochondria and there is calcium here. This is the terminal part of axon. So synaptic cleft is between this neuron and this muscle is known as synaptic. This space is known as synaptic cleft. So the junctional folds of the sarcolemma of, so this is the fold of the sarcolemma of the muscle. So this fold is known as motor end plate. Depression, it's a motor end plate. So this is a part of myofibril of the muscle. So this is the muscle, the outer covering of the muscle. So this is T-tubule. So transverse tubule. So this is known as neuromuscular junction between neuron and muscle. So neuromuscular junction, a nerve signal will arrive at the synaptic end bulb, and this will cause acetylcholine containing vesicle to undergo exocytosis. So exocytosis means this means acetylcholine containing vesicle means exocytosis means this is exocytosis means one of cell vomiting So acetylcholine is released from the neuron with calcium. So calcium is also an important channel. So acetylcholine will diffuse across synaptic cleft and bind to acetylcholine receptor, that is known as nicotinic receptor. These receptors are actually ligand-gated sodium channels. So the binding of acetylcholine causes them to open. Sodium will rush into the cell and making a local cell interior more positive. So this process is known as depolarization. This is a local event. So as I told you, this is nerve area, axon terminal. So this is acetylcholine molecules. This acetylcholine molecules will diffuse across this synaptic cleft and will arise. This is acetylcholine receptor or synaptic receptor. This is acetylcholine receptor. So when the acetylcholine attaches this to acetylcholine receptor, this sodium channel, this is the sodium channel will open. So if the sodium channel will open, so this sodium will rush inside the cell. So if sodium is joining to the cell, the full of the cell will be more positive. So this process is known as depolarization. But also more calcium is in, and if the inner cell is negative, it will be repolarization again. So the very important thing you should keep in mind is characteristics of neuromuscular junction. So this is, we do have 7 characteristics of neuromuscular junction. So neuromuscular junction is the transmission of action potential in neuromuscular junction is unidirectional. So neuromuscular transmission of impulse will go from the neuron to the muscle. So it's only unidirectional. So there is a single neuromuscular junction per muscle fiber. So within the muscle, one muscle fiber will contain one single single nerve. So muscle fiber with nerve will give neuromuscular junction. So single fiber with single neuromuscular junction with single nerve. So the neurotransmitter is always acetylcholine. So it synthesis choline plus acetylcholine. So acetylcholine is by action of choline acetyl CoA transferase. So the storage of this acetylcholine is form acetyl complex with ATP packaged in vesicles. So release by calcium dependent exocytes. So as I told you, acetylcholine will be released from the neuron end plate by calcium dependent exocytosis. So metabolized by action of acetylcholine esterase. So this is an important thing. The post-junctional receptor is always nicotinic receptor. So post-junctional receptor is in the muscle membrane. So that is nicotinic receptor. The effect of acetylcholine on nicotinic receptor is always excitatory. That means excitatory means will initiate muscle contraction by producing end plate potential. So end plate potential has high safety margin. So end plate potential is high safety margin. In general, each muscle that arrives at neuromuscular junction causes 3 times as much of end plate potential as required to stimulate the muscle fiber. The other function is there is synaptic delay 0.3. It is fatigable due to depletion of ATP and neurotransmitter storage. So this is all are very important characteristics of neuromuscular junction. So the other is mechanism of muscle contraction or excitation-contraction coupling. So the process of nerve impulse will go into the end of the axon, that is synaptic end node, so end plate. So the end will stimulate the muscle is known as excitation. So the second step is excitation-contraction coupling. So excitation will be followed by contraction. So when the muscle fiber membrane is depolarized, contraction of fiber follows. So the process by which depolarization initiates contraction is called excitation-contraction coupling. It has several steps as follow. So this all 13 steps is important for muscle contraction. So in general, excitation plus contraction will follow these 13 steps. So the action potential or nerve impulse is initiated and propagated along the motor nerve fiber and arrives at end feet, that means end motor plate. Then after end plate, opening of voltage gated calcium channel in the synaptic interval. So in the synaptic interval, opening of calcium channel. When calcium channel is open, so neurotransmitter will go there, will be removed out. So in the synaptic interval, so plasma membrane, so influx of calcium to trigger release of acetylcholine. So acetylcholine released by calcium dependent exocytosis and diffuse through synaptic cleft and binds to nicotinic receptor on post junctional membrane. So the fourth step is opening of since acetylcholine is attached to its receptor, nicotinic receptor, following it is attachment with nicotinic receptor, ligand gated sodium channel will open and influx of sodium to produce end plate potential, that is high safety margin. So now following sodium joining to the cell, so the cell will be more positive. So the spread of depolarization through sarcolemma or cell membrane. So then spread of depolarization through T-tubule. So depolarization of T-tubule will stimulate sarcoplasmic reticulum. Since sarcoplasmic reticulum is a storage site for calcium, so sarcoplasmic reticulum stimulated then releases calcium into sarcoplasm. So in the sarcoplasm there is troponin C. So calcium binds with troponin C. Troponin C means calcium, site for calcium. So binds with calcium. So now calcium, if calcium is attached with troponin C, this process detaches troponin I from active sites of actin. So if troponin I is detached from actin, so the active site will be free for attachment of myosin. So this is a very important step for muscle contraction. So now after this step 10, the detachment of troponin I from actin displaces tropomyosin and uncovering the active sites of actin filament. Uncovering means the active sites of actin filament will be opened. So now up to this is now still this is excitation. So after this step 11 and 12, 30 is contraction. So when active sites of actin is exposed, the heads of myosin connect to them. So since attachment with troponin C, since it prevents troponin I from the active site, so and also displaces tropomyosin, so active sites of actin is free and head of myosin is attached to it, so making cross bridge between myosin and actin. At this point, myosin has just hydrolyzed ATP into ADP and inorganic phosphorus. So this process will produce energy. So this breakdown takes place by ATPase enzyme on myosin heads. So this will be energy. So this energy is, by the way, muscle contraction and muscle relaxation is an active process, so it needs energy. So this breakdown of ATP into ADP and phosphorus means produce energy. So this energy is important for movement of head of myosin. The movement of head of myosin. So release of energy causes the movement of the head, power stroke, towards the center. This results the sliding of thin filament over the thick filament. So this process is known as contraction. So myosin remains bound to actin until it binds to another ATP. If the head of myosin gets one phosphorus and we form ATP, relaxation process will be occur, then the step will repeat itself. So the myosin remains bound to actin until bound to another ATP. The head of myosin is charged with new molecule of ATP, then detached from actin, this leading to relaxation phase. So normally tropomyosin obstructs myosin binding site of the globular protein actin subunits. So calcium binds to troponin C polypeptide of the troponin triad. This changes the conformation of the troponin, which changes the conformation of the tropomyosin, which exposes myosin binding site on actin. Once actin myosin binding site is exposed, myosin will attach to it. Then at this point, myosin has just hydrolyzed ATP into ADP and phosphorus. However, both molecules are still bound to the myosin. So ATP hydrolysis provide energy for cooking of the myosin head. Once myosin is bound to actin, the myosin head will release ADP into phosphorus, which will cause change in conformation. This results in thin filament slide over thick filament. So this is known as contraction. So after this, the step will repeat itself, and our movement is dependent by this phase. So now, as I told you, the cycle of attachment power stroke is release continuous as long as calcium and ATP remains available. So a common analogy is climbing rope hand over hand. If you are climbing a rope by crossing one hand over the other, this seems like a contraction coupling. So as I told you, this is as we have seen, this is a thin filament, this is thick filament or myosin, this is head of myosin. So head of myosin will power stroke over this because of, since the myosins move just like this, so the myosin is slide over, actin is slide over myosin. So due to this myosin head will move. So this will give energy. So this is how muscle will be contract, or how myofibril or sarcoma will contract. So the other is contraction. So as I told you, contraction is this from 11 to 30 step is contraction phase. But to give you some separation, this is a contraction phase. So once actin myosin binding site is exposed, myosin will attach to actin. At this point, myosin has just hydrolyzed ATP into ADP. So we have seen it. So this is process known as contraction. So this picture will shorten the steps from 13 to 8. So this is nerve terminal or nerve ending. Step 1 is axon terminal of somatic motor neuron is this one. So this is acetylcholine. So acetylcholine will move or will diffuse by calcium dependent exocytosis here. So this is a muscle membrane. So by this time this is sites, that is these sites are known as that is site for or receptor for nicotinic receptor. So by this phase step 2 action potential will move in this way, and will go into, this is muscle fiber, so we'll go into T-tubule or transverse tubule. In the transverse tubule there is DHP receptor. So by that time as I told you, this area, this is the nicotinic receptor. When acetylcholine arrives in the nicotinic receptor, this sodium channel will open, so sodium will enter. So sodium is positive, so positive will join. So by this time this is motor end plate. So this is sarcoplasmic reticulum. Sarcoplasmic reticulum is the site for here calcium storage. And by this time in the T-tubule, so neurotransmitters also will join here. So step 3, after more sodium is joined into T-tubule and sarcoplasmic reticulum, this calcium is released from if more sodium is joined, so calcium is this site is sarcoplasmic reticulum. So from sarcoplasmic reticulum, so calcium will move into the cytoplasm or sarcoplasm. So in this area, calcium is freely moved in the sarcoplasm. So finally, in the step 6, calcium binds with troponin. So this calcium, this screen is attached with this troponin molecule. So if it is binded here, so in this case the tropomyosin uncovers binding site. So this is a tropomyosin. So tropomyosin will expose or uncover the binding site. Then having this concept, then myosin head execute power stroke. So by that time, calcium troponin attachment will prevent troponin I. So troponin I will make it tropomyosin uncover. So by this process, the myosin head execute power stroke hit. Myosin head is making will break down into ATP into ADP and phosphorus, so will be produce power stroke. So this is thin filament. So thin filament is slide over this thick filament. So the thin filament, this is head of thick filament. So finally, actin filament moves toward M line. So actin filament is this one. Actin filament is more move. This is M line. So actin filament is moves toward the M line. So this is the distance. So actin filament moves from 2 to up to this distance, So this is the process of muscle contraction. So another picture is here. The step 1 is a resting fiber. His cross bridge is not attached to actin. So this is a thin filament. So this is thick filament. So this is thick filament. In this way, here is ADP. This is phosphorus. This is a cross bridge, and this is myosin head. So in step 2, cross bridge binds to actin. So this cross bridge is binding to actin. This one is not binded. In this case, phosphorus, inorganic phosphorus, is released, then causing conformational change in myosin. Then power stroke causes filaments to slide. So ADP is released. Finally, a new ATP binds to myosin head, allowing to release from actin. Then this is ATP. If ATP is formed again, ATP is hydrolyzed, causing cross bridge to return to its original orientation. So this is relaxed. So this is another step. So mechanisms of muscle. Now up to now we are completed step 3. Now it is the last step, that is muscle relaxation. To be muscle relaxed, it will be follow the following steps. 1 is action potential must cease for muscle to relax. So if there is action potential or impulse, if there is impulse or action potential, that will stimulate the muscle, so there will be relaxation. So to be relaxed, there is action potential is must stop for a while. The other important step is calcium should be reuptaken back into sarcoplasmic reticulum from sarcoplasm by calcium pump. As I told you first, as you know, calcium is the storage site in calcium is stored in sarcoplasmic reticulum. So as I told you before, to increase muscle contraction, calcium is released from sarcoplasmic reticulum to sarcoplasm, then binds to troponin C. Now it should be calcium is separated from troponin C and will move from sarcoplasm into sarcoplasmic reticulum. That's the original place that is known as calcium pump. When the muscle ceases being stimulated, the calcium pumps win. The sarcoplasmic drop, meaning calcium will drop in the sarcoplasm and will increase in the sarcoplasmic reticulum. So decreased calcium in the sarcoplasm or cytoplasm, calcium detaches from troponin C. If troponin C is detached, tropomyosin will be covered the active sites of actin. That means before tropomyosin is uncovered, now tropomyosin is covered the active sites of actin. So if tropomyosin covers the active sites of actin, so there is no active site place for myosin head. So for head of myosin charged with ATP and detached from actin. So head of myosin charged with ATP and detached from actin. So by this time muscle relaxation is occurred. So muscle relaxation, an active process, it's requiring energy. Now a large amount of energy is consumed during muscular performance. One is ATP or energy is important to move the head of myosin or power stroke. Active calcium pump to move calcium from sarcoplasm to sarcoplasmic reticulum, and for sodium potassium pump in the membrane. For 3 things we use ATP or energy. So having this concept, now let us see types of muscle contractions. Muscles can be contraction can be isometric, the same measurement, or isotonic, the same tension. So let us see isotonic contraction. In the isotonic contraction, there is no change in muscle tension, but there is change in length of the muscle. That means there is shortening of the total length of muscle. In this case, work is done. In this contraction, work is done. So this occurs when the contraction moves an object to moderate weight. So this is length of the muscle. So when it contracts, it will be short. In this case, we can say isotonic contraction. In this case, work is done. But in the isometric contraction, there is a change in length. Occurs when muscle contraction fails to move heavy objects. In this case, there is no work done. In this type of contraction, there is no work done. So this is the length is the same. So this is known as isometric contraction. Another important concept is muscle tone. Muscle tone is a constant, slightly contracted state of all muscles. This does not produce active movement. Keep muscles firm, healthy, ready to respond to stimulus, and results from low rate of nerve impulses. So essential for maintaining posture or head upright. So this is known as muscle tone. So muscle tone makes our body in slight contracted state, and our muscle will be ready for next stimulus. So the other when the muscle is became out of normality, it will be hypertrophy and atrophy. Hypertrophy is a process in which an increase in size of cell, tissue, an organ is called hypertrophy. Hypertrophy will occur when there is more synthesis of myofibrils, or increase the number of myofibrils is a reason for occurrence of hypertrophy. So in contrast, a reduction in size of cell, tissue, and organ is called atrophy. This atrophy is mainly will result due to disease activity. So if we are not using one muscle, that muscle will be reduced in all aspects. So that is will result in wasting. So this decrement of size is called atrophy. So now we have completed skeletal muscle. Now it is site of smooth muscle. Now this smooth muscle is will get in the next video, so don't forget to subscribe this channel, so you will get the next videos. So have a nice time. Bye bye.